viral exanthems. Okay. So, what are the learning objectives for today's lecture? To that, at the end of the session, the student should be able to understand and enumerate the causes of viral exanthematous infections, etiopathogenesis of these infections, and we'll be discussing 4 very important viral infections that are measles, chickenpox, smallpox, and rubella. Okay, many of the other viruses which also cause exanthems have already been covered in other topics or would be covered subsequently. But these 4 main viruses will be covered in today's lecture. So what is an exanthem? Exanthem is any eruption or rash on the skin, and that may be or may not be associated with fever or other systemic symptoms. Okay, so majority of the exanthems are infectious, and many of them or most of them are caused by various viruses. And exanthems can also be due to various drug reactions or some allergic manifestations to drugs or some other compounds. Okay, but we'll be dealing only with exanthems which are caused by various viruses. So this is the list of various viruses that are capable of causing exanthems or any other form of skin lesions in human beings. So the important ones include herpes virus, the entire group of herpes virus, namely HSV 1 and 2, Variola Zoster virus, which causes chickenpox and zoster, Epstein-Barr virus, and human herpes virus 6. Out of the entire group, these 4 or 5 viruses can cause viral exanthems. Then herpes virus 6 causes roseola infantum, which is also known as exanthem subitum or the 6th disease. Then there are other DNA viruses which include pox virus such as smallpox and molluscum contagiosum, parvovirus which causes erythema infectiosum, which is also known as 5th disease, and then we have HPV or the human papilloma virus, which causes various forms of warts or condyloma acuminata. Then we also have certain RNA viruses like Measles virus, Rubella virus, Coxsackie virus, and sometimes even the arboviruses such as dengue, Ebola, and Ebola is not an arbovirus, but similar viral hemorrhagic fevers, including arboviruses and other viruses, can cause viral exanthems. Then you have heard this term, what is 5th disease, what is 6th disease. So what are these diseases? So these diseases are a list of 6 diseases which was important historically, and these diseases used to occur mainly in the childhood. So it included measles, which was the first disease, scarlet fever, then rubella, Duke's disease is a question mark because later on it was found that probably this is a disease which is very much similar to scarlet fever. So Duke's disease is not a verified entity. It was supposed to be or thought to be caused by Staphylococcus, and hence it was known as Staphylococcal Scarlet Skin Syndrome, SSSS, which is a toxin-mediated disease. Then 5th and 6th disease are the erythema infectiosum and roseola infantum. So these terms, 1st, 2nd, 4th, 5th diseases are nothing but the number at which they occur in the list of the disease. Then coming to herpes virus infections. So herpes virus has been taught to you in STI, I think, mainly. So we'll be covering only the varicella zoster part of this group. So these viruses, they include a group of viruses that have the unique property of being able to establish latent infection or persistent infection in the host in various cells of the body, and subsequently they can undergo periodic reactivations. Morphologically, they are spherical, they have equicidal symmetry, and their structure, as you can see in this diagram, they have a viral genome that is double-stranded DNA enclosed by a nucleocapsid. Then outside there is a tegument followed by envelope, and from the envelope there are projecting envelope proteins which are glycoprotein in nature. So this is the basic structure of the herpes virus, and the structure is more or less similar for all the members of this family. All right. So then there are essentially 8 human herpes virus named from HHV1 to HHV8, and they are placed in various subfamilies alpha, beta, and gamma, and they are able to establish latent infection in various organs or tissues of the body. So HHV1, 2, and 3 are HHV1, 2, and Varicella Zoster virus. So Varicella Zoster virus that we'll be discussing belongs to the alpha subfamily, able to establish latent infection in neurons, and its official name is human herpes virus 3. Then we also have CMV, HHV6, HHV7, Epstein-Barr virus, which is HHV4, and then HHV8 is also there, which is causing Kaposi sarcoma virus. So this is a very important classification for human herpes virus. So like I told you earlier also, we'll be discussing only Varicella Zoster virus among all the herpes viruses, because other members have been covered or will be covered. So this Varicella Zoster virus is an important cause of viral exanthems, and it causes 2 very important clinical entities. The first one is known as chickenpox, and second is the herpes zoster or shingles. Okay, so chickenpox is the primary infection, and zoster or shingles is the thing that is occurring as a result of reactivation of the latent infection. So this is this virus, the Varicella zoster, belongs to the alpha herpes virus group. Again, it has double-stranded DNA, and only one serotype is known. Okay, although there is some cross reaction with HSV virus. So epidemiologically speaking, primary varicella or the chickenpox, it is an endemic disease. Doesn't usually occur as epidemics. It is an endemic disease. That means it is present in some number throughout the period, and it is one of the classic childhood diseases with very high prevalence in the 4 to 10 year old age group. It is highly communicable with very high attack rate of 90%. That means if 100 people are exposed to a child suffering from the disease, up to 90% can develop the infection. So one attack, it confers long-lasting immunity, and humans are the only reservoirs and only species which can harbor this virus, and there is no carrier stage. Herpes zoster, in contrast, it occurs sporadically and unevenly throughout the year, mainly in people who have been infected with this virus some time back, and the virus has established itself latently in the neurons of that person. So chickenpox, it enters through upper respiratory mucosa. So the route of transmission is aerosol and contact transmission. From the local site, it spills over to blood and is carried through the infected mononuclear cells to target sites such as skin, respiratory tract, from where it is shed in the respiratory secretions, and neurons. So this virus is able to undergo latency in the neurons. So typically, the incubation period is approximately 2 to 3 weeks, and the rashes that appear in chickenpox are vesicular in nature. Vesicular means they are small fluid-filled lesions that sometimes appear like dewdrops on the skin. The distribution is centripetal. That means it involves the central part of the body, that is the trunk and the neck and the face, and limbs are relatively less involved. The lesions are bilateral and they appear in multiple crops. That means the lesions, they appear in groups, and when the previous group of fresh crop of lesions, they again appear, and fever appears with each fresh crop of the rashes. Complications include secondary bacterial infections of these vesicular lesions. Then CNS involvement can be seen in form of encephalitis or aseptic meningitis in children, but most serious complication is varicella pneumonia, which is more commonly seen in adults. So these are complications. Otherwise, the disease itself is self-limiting, but if the complications develop, then it can get relatively serious. Then Ray syndrome can be seen, in which the liver and the CNS is involved. Other complications include myocarditis, nephritis. But in pregnancy, chickenpox can affect both the mother and fetus. So an expecting mother is at high risk of developing varicella pneumonia, and the fetus itself is at higher risk of developing congenital varicella syndrome. All right. So when a mother is pregnant, the fetus can develop two types of diseases. One is the congenital varicella syndrome, and another is the neonatal varicella. So congenital varicella syndrome occurs if the mother is suffering from varicella or chickenpox during the earlier part of the pregnancy. That means within first 20 weeks of the pregnancy, when the fetus is still developing. So the child that is born to such a mother may develop congenital varicella syndrome, that shows scarring of skin of the newborn, hypoplasia of the limbs, various forms of defects of the CNS and eye, and even death can be seen, death of the child. On the other hand, neonatal varicella is seen if the mother becomes infected with this virus near the time of delivery. Okay, so the varicella zoster virus can cause the placenta in the very late stages of pregnancy and infect the virus congenitally. If the rash appears in the mother more than 1 week before the delivery, then sufficient immunity would have been transferred to the fetus, and it is not likely that the child would suffer. So, if the mother is developing rashes within 1 week of delivery, all right, that means up to 7 days prior to the date of delivery, if the mother is developing chickenpox, that is primary varicella, then it is likely the fetus or the newborn may suffer from neonatal varicella. If the mother has got the disease before 1 week of the delivery, prior to the delivery, then it is unlikely that the neonate will get this disease. All right, then second entity is the zoster or shingles or zona. This is mainly due to reactivation of the latent varicella zoster virus in the old age, mainly in immunocompromised individuals or occasionally in the healthy adults. This disease, as I told you earlier, the virus remains latent in the neurons or the ganglia, sensory ganglia, and intermittently it reactivates, resulting in a painful disease. But this disease, it restricts itself to a particular dermatome. That means a dermatome, as you know, is the area of the skin that is innervated by one particular group of nerve. Okay, so any sensory ganglia which is involved by this virus, this disease would appear only in the skin that is supplied by that ganglia. So the symptoms, they typically start appearing along with pain in the affected dermatome, and it is followed after 2 to 3 days by a vesicular eruption. Okay. So as you can see, it can occur in the thoracic area, or it can occur in the facial area. So facial area, mainly the more commonly the area that is supplied by the trigeminal nerve, the ophthalmic branch of the trigeminal nerve, and sometimes the thoracic nerves, the area supplied by a particular thoracic nerve, are commonly infected sites. So it basically restricts itself to a particular dermatome. Then there can be some complications of zoster, which include post-herpetic neuralgia, that is neural kind of pain that persists even after the rashes have gone. Zoster ophthalmicus, which basically involves the eye along with the other parts of the face, especially if the trigeminal nerve is involved. Then if the facial nerve is involved, then Ramsay-Hunt syndrome, it can be seen, in which there are painful rashes, and it may even cause facial nerve paralysis and hearing loss in the affected ear. Very rarely it can also involve the internal visceral diseases. So this is a patient suffering from Ramsay hand syndrome with facial nerve paralysis following zoster. So for diagnosing chickenpox or varicella zoster virus, most of the cases are generally diagnosed clinically, and very less commonly laboratory support is required. But the specimen that can be collected include vesicular lesions or the scab or the macropapillary lesions, the secretions or exudates from that. First, uh, first thing that can be done is preparing a Giemsa staining from the scraping of the ulcer base. Okay, this is known as Zank smear. So you basically scrape the vesicle or the ulcer and take the exudate, spread on a slide and stain with Giemsa stain. So you are likely to see multinucleated giant cells, which are also seen in other HSV infections. The virus can be isolated in various cell lines, in which it can produce HSV-like cytopathic effects. So this is a Zank smear, a slide that is prepared from exudates or secretions from the base of the ulcer and stained with Giemsa stain, showing presence of a multinucleated giant cell. So this is Zank smear, frequently done for HSV infections also. Then various forms of ELISAs and PCR are also there to diagnose varicella zoster infection. For treatment, we have certain antiviral drugs which can basically be used to prevent complications of chickenpox and also halt the progression of zoster in adults. So these include acyclovir, famciclovir, valacyclovir. All these are various antiviral drugs. Vaccine is also available now, very frequently being given in children in India as well. So it is a live attenuated vaccine that uses OKA strain of the varicella zoster virus. It is given to children after 1 year of age. 2 doses are given at the 12 to 15 month, and then second dose is given at 4 to 6 years. So chickenpox vaccine is now very frequently given, especially in the private sector. It is not part of the national immunization schedule, but it is given very frequently nowadays. Then we also have varicella zoster immunoglobulin, that is a part of passive immunity. Okay. It is usually used as a measure for post-exposure prophylaxis, and it has to be given preferably within 72 hours of exposure. So these are nothing but immunoglobulins that are specific for varicella zoster virus. So it may be given in certain situations where adults who are at higher risk of varicella deaths, or immunocompromised patients like HIV, or women during pregnancy when they have been recently exposed to this disease. So they can be given varicella zoster immunoglobulin or VZIG to prevent the disease from developing in them as emergency prophylaxis. It can also be given in neonates born to mothers suffering from chickenpox if the mother developed chickenpox very close to the date of delivery. Then, since it is a airborne or aerosol mediated disease, so all the airborne precautions must be taken, and direct contact with the patient must be avoided. The patients must be kept in isolation. Then the second important group of viruses are the pox virus. So pox virus are the largest virus among all the viruses that are capable of infecting human beings. They are approximately 400 nanometers by 230 nanometers in size, and they are so large that they can be seen under light microscopy if you look carefully. They are quite complex viruses. Their structure resembles that of a brick. Okay, so externally there is an envelope, and it encloses a core in the center part, and on the sides there are two lateral bodies whose function are relatively unknown. So the core or the nucleocapsid, it is dumbbell shaped, biconcave dumbbell shaped. It also possesses double-stranded DNA, and it is the only DNA viruses that can replicate in the cytoplasm of the host cell. So pox viruses include Variola virus, which is a causative agent of smallpox, Vaccinia virus. So Vaccinia virus is a separate virus that is antigenically somewhat related to the smallpox virus or the Variola virus, and this Vaccinia virus is used as a vaccine. Modified Vaccinia virus is used as a vaccine for smallpox. And another virus is Molluscum contagiosum virus, which causes wart-like lesions, Molluscum contagiosum. So talking about smallpox, as you all know, smallpox was the first disease that has been eradicated from the world, and it used to be fatal. And out of around 10 people who contracted the disease, many of them died. And those who survived, they had scars, very disfiguring kind of scars were there, mainly on the face. Okay, so this was a severe disease that disfigured people, and it also led to death of many people. So the last natural case of variola major was seen in a Bangladeshi woman who was found in Assam on 24th May 1975. And when this last case was discovered, that is towards the end of eradication of the smallpox, immediate steps were taken, and within an area of 1.5 kilometer radius, everybody else was vaccinated immediately, so that anybody who has been exposed may not spread or may not develop the disease. And last natural case of variola minor was seen in Somalia in 1977, and subsequently the disease was declared as eradicated from the world by WHO after 3 years of the last 1980. So this is the criteria that WHO usually follows, and even for polio, they are following the same criteria, that after 3 years have elapsed after the last case being reported, the area is declared free from that particular disease. So it was eradicated in 1977, but in 1978 there was a small outbreak in Birmingham due to the accidental release of the virus, following which most of the stocks that were kept in laboratories were destroyed, and presently only in USA and Russia they have the stocks of this virus, mainly for scientific research purposes. Otherwise, the virus has been destroyed from all the other laboratories that were keeping them. Since it has been eradicated, and now for last 20-30 years people have not been exposed to this virus, so this poses a threat for bioterrorism, because people after 1980s, they have not been exposed to this virus and they don't have any sort of immunity to this virus. So it is a very potential agent for bioterrorism. So what are the reasons that made the eradication successful? First was that it was an exclusively human pathogen. There were no animal reservoirs. There were no environmental reservoirs. So if you vaccinate human beings only, this disease could be eradicated. And patients were the only source. There was no carrier stage. There was no long-term carrier stage in human beings also. Case detection was easy because of characteristic appearance of the rashes. Anybody who developed rashes similar to that seen in smallpox could be identified easily. Subclinical cases, they were not transmitting the disease. This global eradication program was launched in 1967, and within around 10 years, there was the disease was eradicated. Other important factors that played role in eradication were the vaccine was highly effective, and it was Vaccinia virus that was used. This vaccine was different from the one Edward Jenner had devised. This vaccine was live Vaccinia virus, and it was freeze-dried form. That means lyophilized form, in which could be stored at normal temperature. Didn't require any special storage conditions. And the technique of vaccination was also very simple. Didn't require any special training or was not very technical. It basically consisted of a bifurcating needle, which carried the drop of the vaccine, and multiple punctures were done on the skin using that. So if you see people from before 1980s, many of them, including our parents, they have this scar on their right shoulder. So this scar is that of the vaccination done for smallpox, because after vaccination there was a small ulcer and pustular kind of lesion which healed with a scar. So this scar on the right shoulder is indicating smallpox vaccination, and another scar that we have on the left shoulder, that is generally for the BCG vaccination. So this is the needle that was used for giving smallpox vaccine. It is a bifurcated needle, and between the two prongs there's a drop of the vaccine. So this needle was held in one hand and it was pricked into the upper part of the arm multiple times. So this is how the Vaccinia virus Vaccinia vaccine was delivered. So clinical manifestation of this disease was again mainly spread through the respiratory tract. Incubation period was short, and the rashes were different from that we have seen in the chickenpox. These rashes were deep-seated, involving deeper parts of the skin and subcutaneous tissue, and various stages of these rashes were seen. Initially, the rashes would be macular. That means that they were not raised, they were at the level of the skin, then gradually becoming papular, which are slightly raised, solid lesions, then vesicular, which are fluid-filled lesions, pustular, which tend to become purulent, and finally they healed with scabbing and scarring. The distribution was centrifugal. That means involvement of peripheral areas, not mainly involving the trunk, like the areas such as face and limbs were preferably involved, and fever subsided with the appearance of rash. So this is a typical case of smallpox. Vaccination may, like I told you, the vaccine that was used for eradication of smallpox was based on the Vaccinia virus. But before that, a practice that was known as variolation was quite common, and it was the initial attempts to provide artificial immunity against smallpox. So what was done basically, any person who was suffering from smallpox, his skin scrapings were inoculated onto healthy people. So basically, people were being infected deliberately with this virus in a hope that they would develop immunity. So this practice was not actually very right, because you are giving infectious agent that is very much capable of causing disease to other people. But the fear of this disease, that the death was so much that they felt that variolation or infecting a person with lesser dose of virus into the skin would lead to fewer complications, lesser fatalities, and lesser rashes. So this practice was being followed till 17th or 18th century. After that, as you all know, Edward Jenner came up with cowpox vaccine. So this vaccine was also somewhat similar to the practice of variolation, but the difference was he used a different virus, that was cowpox. So what he had observed was that women who were milking cows that were suffering from the disease that is known as cowpox, and the woman would also suffer from this disease, cowpox disease, but it was milder form, and then subsequently they would not develop smallpox. So he used this extract from the lesions and injected into द सन ऑफ हिज गार्डनर, ठीक है, उसका जो गार्डनर था उसके बेटे में उसने इंजेक्ट किया, एंड सब्सिक्वेंटली ही आफ्टर फ्यू मंथ्स ही इन्फेक्टेड हिम विद smallpox virus, but he did not develop the disease. That is how this vaccine was developed by Edward Jenner. But the vaccine that was used was live Vaccinia vaccine. Okay, it was a different virus, as you know, Vaccinia virus, and it was given as a single dose after 1 to 2 years of age by scarification. So Vaccinia virus, it cross-reacts with variola, and the antibodies produced against Vaccinia are also protective against variola. Okay, so this antigen, this cross-reactivity was so much that it was able to eradicate the variola disease, that is a smallpox disease. And the disease or the virus itself, the Vaccinia virus, it was relatively non-pathogenic for human beings or produced only mild skin lesions. Then another virus is the Molluscum contagiosum, which is an obligate human pox virus with characteristic skin lesions. So इसके skin lesions जो होते हैं, they are relatively smaller in size, around 2 to 3 mm in size, but they have very typical appearance, in a sense that they are pearly white wart-like lesions that are umbilicated. That means lesion ke center mein there is a dimple, or there is a like an umbilicus, there is a depression in the center of the lesion. So this is the typical appearance of Molluscum contagiosum. This disease, Molluscum contagiosum, is mostly sex-limited, but in patients with HIV or other immunocompromised patients, the disease can become more generalized and severe. Okay, so the next virus that we are going to talk about are the myxovirus. So myxa means mucus or mucin, mainly because they have affinity for certain mucinous substances. They are able to cause agglutination of the red blood cells, RBCs. If you mix these viruses with RBCs, then the RBCs would clump together, or they would get agglutinated because of presence of the specific receptor on the virus, specific protein on the virus that links the mucoprotein receptor on the RBCs. Okay, so myxoviruses are broadly divided into orthomyxoviruses, which are the influenza virus, and the paramyxoviruses, which includes other viruses like the parainfluenza viruses, measles, mumps, all those things. So respiratory viruses have been covered. We'll be dealing mainly with the measles virus in the paramyxovirus group. So these paramyxoviruses are pleomorphic in nature. That means they can form long filamentous or giant forms. They can be up to 300 nanometers in size. They are single-stranded negative sense RNA viruses. But unlike orthomyxoviruses, the genome of these paramyxoviruses are unsegmented. That means there is a single segment of the genome, and because of which they do not go genetic recombinations or antigenic variations, which are very frequent in the influenza virus, like you know. Okay, so paramyxoviruses are antigenically stable in contrast to the orthomyxoviruses. So on the left side, the purple thing that you see is the orthomyxovirus, and on the right side, the green one is the paramyxovirus, typical representative figures. So you can see that in the orthomyxovirus, the genome is segmented into 8 segments, which facilitates genetic recombination. But in case of orthomyxovirus, there is a single segment. So this is electron microphoto of paramyxoviruses showing pleomorphism with various filamentous forms. Then orthomyxoviruses, like you know, they have matrix protein or the M protein, hemagglutinin, and neuraminidase, HNN glycoproteins. Okay, but the paramyxoviruses have similar glycoproteins, but they are slightly different. So they have M protein like the orthomyxoviruses, but instead of H and N, they have HN component or only the H component. So HN is present only in a single glycoprotein, and H, that is the heme agglutinin, is maybe present in place of HN, and these are responsible for heme agglutination and elution. Then they also have another protein, that is the F protein or the fusion protein, which promotes the fusion of the virus with the host cell, and it also permits fusion of various host cells together, resulting in formation of giant cells or syncytia formation. So this is a typical paramyxovirus. So this is showing what is heme agglutination. So if RBCs are left on their own in a test tube, they would settle at the bottom in form of a small dot. Okay. But if they are mixed with virus, which is capable of causing heme agglutination, like the orthomyxovirus or the paramyxoviruses, then they would cause heme agglutination, and at the bottom of the test tube or the microtiter well, it would form a carpet kind of thing. Okay, so because the RBC, they are agglutinated or clumped together by the receptors or the proteins which are present on the viruses. But if you add specific antibodies, okay, against these viruses and then add to RBCs, then these antibodies would obviously bind the virus, and RBCs would remain free and they would not agglutinate and again settle at the bottom. So this test is known as heme agglutination inhibition. Okay, so heme agglutination test, that the B row would indicate that there is presence of virus, and heme agglutination inhibition test is basically used to detect antibodies in the patient's serum, or to detect if we already has very specific antibodies, then we can detect virus कि कौन सा virus है. Suppose we want to detect कि measles का virus है या parainfluenza virus है, so we would take specific antibodies against that parainfluenza virus, which we know parainfluenza virus, and then we test virus with RBCs and the known antibodies, and if the heme agglutination is inhibited, then it would indicate कि जो antibody थी हमारे पास, which is known to us, has bound to the virus and inhibited heme agglutination. So this is the virus is the one for which we have the antibody. Okay, so this is the role of F protein or the fusion protein, which allows the entry of the virus, and same F protein, when the virus enters into the cell, it is expressed on the surface of the host cell also, the epithelial cells. So these cells, the host cells, they fuse together, resulting in formation of syncytium or the giant cell. So this is a giant cell seen in the your cell lines, various cell lines, pure cell lines or other cell lines. Okay? Syncytium formation is typically seen here. So we have large number of paramyxoviruses, parainfluenza viruses, mumps, measles, respiratory syncytial viruses, human metapneumoviruses, but others have been covered. We'll be talking only about the measles virus, which is very important cause of viral exanthem. All right, so measles. So measles is one of the 5 or 6 classic childhood viral exanthems along with other diseases. It is a RNA single-stranded negative sense RNA. Outer proteins include the M or the matrix protein, fusion protein, and the H or the heme agglutinin component, but there is no neuraminidase activity. That means there is no elution. Elution means after some time, when the RBCs have been agglutinated, elution takes place. That means the viruses, they are released from the RBCs, and again the RBCs can the agglutination gets over and the RBCs settle down in form of a dot. That is elution. So this is the structure of measles virus with H protein and the F protein, heme agglutinin, and the fusion protein. So natural infection is seen only in human beings. Mainly children are affected. Airborne transmission is there with very high infectiousness. It is highly contagious disease, and 90% secondary attack rate is seen in susceptible individuals. Okay, there is a single serotype, and immunity is permanent after a person acquires disease. Incubation period is around 1 to 2 weeks. So after the virus gains entry through the respiratory tract, it multiplies locally in the respiratory epithelial cell, and then subsequently it spreads to regional lymphoid tissue where further replication occurs. Then there is primary viremia that disseminates the virus into the various organs of the body. It spreads mainly to the reticuloendothelial cells where it replicates, and subsequently there is a secondary viremia which then subsequently seeds the epithelial surfaces of the body, including skin, respiratory tract, conjunctiva, where focal replication subsequently takes place. So this is how the measles disease, measles virus gains entry and spreads and establishes itself in the human body. Okay. So initially there is a stage known as prodromal stage in which the person feels general tiredness, malaise that lasts for around 2 or 3 days, followed by high fever, cough, coryza, conjunctivitis, and during this time when the person is having cough, the disease is most infectious. So exanthematous lesions include complex spots which are not present on the skin, but they are present on the mucous membrane of the oral mucous membrane, typically seen on the buccal mucosa. That means mucosa of the cheek near the lower molars. Okay, so these are known as complex spots, typically seen in measles. And then on the skin, reddish maculopapular rashes are seen, and they follow a typical sequence, initially appearing behind the ear, then they spread along the hairline, involving the face, neck, chest, back, abdomen, limbs, hand and feet. Okay, so this is the sequence that the rashes of measles follow. So these are the complex spots. These reddish dots that you can see over here are the complex dots that appear on the buccal mucosa near opposite the lower molars. And then this is a child suffering from measles with typical maculopapular rash. Coryza, somebody is asking what is coryza. Coryza is nothing but flu-like symptoms like runny nose and all those things. Okay? That is coryza. Okay, so measles rashes is one thing, but what we are afraid of are the complications that follow measles symptoms. Okay, secondary bacterial infections, pneumonia, otitis media can be seen, myocarditis, bronchitis, or croup-like features. All these things can be seen. Respiratory or serious manifestations can be there, but the neurological complications, although rare, they are very rare, but they can sometimes be seen after measles infection, many years after the initial infection. So there are two groups of neurological complications. Okay. One is known as post-infectious encephalomyelitis, also known as ADEM, or the acute demyelinating encephalomyelitis. Okay, ADEM. It is seen in one patient out of 1,000 patients suffering from measles, and it is an immunological reaction. Second one is SSPE, that is subacute sclerosing panencephalitis. It is even rarer, and it occurs after 5 to 15 years of the infection, mainly because of defective viral replication, which is unable to produce functional M protein. So the virus, it remains latent for very long period. There are very high antibody titers in the CSF and serum. So these are the two important neurological complications of measles virus, ADEM and SSPE. So again, lab diagnosis is generally not required frequently. The disease can be reliably diagnosed on clinical grounds, and lab help may be required only in atypical cases. Okay, so antigen detection can be done in epithelial cells from the respiratory secretions in urine. Okay, gene staining can help in revealing the giant cells. For isolation and identification of the virus, nasopharyngeal or the conjunctival swabs, blood, respiratory secretions, and urine can yield virus. And cell lines like monkey or human kidney cell lines can be seen. And the cytopathogenic effects typically that are seen include formation of multinucleated giant cells, again because of presence of the F protein or the fusion protein, which helps in syncytia formation. There are also presence of intracytoplasmic inclusion bodies. Here, fluorescent tagged antibodies can help in detecting measles antigen in the cell line. So if you want to identify which virus is actually there, because multinucleate giant cells can be produced by many viruses in the cell lines. Okay, which virus is causing that? That can be done using specific antibodies against measles viruses that are tagged to fluorescent tag. So if if the virus that is multiplying in the cell lines is measles virus, then these specific antibodies would bind to that measles virus, and fluorescence would be seen under microscope. Okay, but if it is not measles virus, then these specific antibodies will not bind, and there would be no fluorescence seen in the cell lines. Okay, so this is how we can diagnose the disease. Apart from that, serological diagnosis, in which we perform ELISA or some other similar tests like heme agglutination inhibition or neutralization test can be done to detect four-fold rise in antibody titer between two samples collected subsequently, or demonstrate presence of IgM, which would indicate recent infection. Okay, management is general, no specific antiviral drug is available for this. So general rest, nursing, symptomatic therapy for fever. Vitamin A is supposed to play a supportive role in measles. So vitamin A is also always given in patients suffering from measles infection. For prophylaxis of measles, we have a well-established vaccine, that is measles vaccine, that is either given alone as measles vaccine, or it can be given as MR, which includes measles and rubella, or MMR, measles, mumps, and rubella. So the strain that is used most frequently is the Edmonston Zagreb strain. It is attenuated by passage in the human diploid cell lines, and it is a live attenuated vaccine. So this strain of the virus is live. Okay, that means the virus is alive, it is not dead, and it is attenuated. That means it can elicit antigenic or immunological response, but it is not capable of causing disease in human beings. It is attenuated or weakened. Okay, so this vaccine is given in 2 doses subcutaneously after 9 to 12 months. So measles was very well in control, but few years back there was large outbreak of measles in US. Okay, that was mainly because of people becoming complacent. They thought that measles is not a disease that is seen in US, so we should not be taking vaccine against measles. And as a result, the cases of measles went up in US until it was declared an emergency situation. So the another virus is rubella. Okay, its vaccine is also given along with measles and mumps. So MMR, it includes measles, mumps, and rubella. But rubella doesn't belong to paramyxoviruses. It belongs to a separate family of toga virus or toga viridae. And toga viridae has multiple genera, and the ruby virus genera, it includes only one member, that is the rubella virus. Okay, so it is a distinct virus. The disease it causes is known as German measles, or rubella or German measles. It is a mild exanthematous fever characterized by macular rash and lymphadenopathy. But it is not because of these rashes that this disease is important. It is important because of its teratogenic effects. Okay, that means the effects it causes in the fetus if the mother becomes infected with this disease. So this disease, rubella, is a part of a group of infections that are collectively known as torch group of infections. Okay, so these torch group of infections are infections which are very capable of causing teratogenic effects. Hence, whenever a woman is expecting a child, we always test for this group of infections to rule out possibility of uh, the fetus becoming infected. So TORCH, it stands for, T stands for Toxoplasma, R for Rubella, C for CMV or the cytomegalovirus, and H for Herpes virus. And O may sometimes is treated as others, and important other infection is the syphilis. So these are the 4 or 5 diseases which are always tested for in a pregnancy. So humans are the only host for rubella, and it is transmitted again by the respiratory route. Incubation is again 2 to 3 weeks. And in normal adult healthy patients or even children, healthy children, uh, a generalized rash appears first on face, then spreading to trunks and limbs. But the rash is generally discreet and it generally disappears by third day. It is not a very severe kind of rash. Sometimes lymphadenopathy can develop and rare complications like arthralgia or purpura or encephalitis can be there, but they are rarer complications. Again, like I told you, this disease is important only because it can cause congenital disease, teratogenic effects, and this is known as congenital rubella. So this virus can result in chromosomal breakages and inhibition of mitosis in the infected embryonic stage. Okay, so fetal damage caused by mother suffering from active rubella is related to the stage of pregnancy. Okay, agar early pregnancy mein the mother gets infected, then it would result in death of the fetus. And if as the pregnancy progresses and the mother becomes infected during the later part of the pregnancy, the damages they become more and more subtle because the development of fetus has already taken place. Classical congenital rubella syndrome is seen in the fetus, which includes cardiac defects, cataract, and deafness. So these are the classical triad: cardiac defects, cataract, and deafness seen in neonates with congenital rubella syndrome. Okay. So this is a newborn suffering from cataract. As you can appreciate in the eyes of this baby, the lens it is opaque, signifying cataract. Okay, diagnosis of rubella virus culture can be done, but again it is not routinely done. Serology is what is done routinely, but it becomes essential to be able to identify whether the expecting mother is suffering from active rubella or whether it is old disease. So we have to be able to identify कि एक्टिव रुबेला है या फिर पुरानी एंटीबॉडीज बनी हुई है मदर के अंदर, बिकॉज अगर पुरानी एंटीबॉडीज हैं देन द न्यूनेट और द fetus is not at risk. ठीक है, the fetus or newborn is at risk if the infection is recent and current and active. ठीक है, so you have to be able to distinguish between them. So for that we have IgM antibodies which indicate recent infection and IgG ability test. That means IgG antibodies if they are detected, then IgG ability test is done. ठीक है, जिसमें हम देखते हैं कि IgG एंटीबॉडी और एंटीजन के बीच में जो स्ट्रेंथ है, बॉन्ड का स्ट्रेंथ है वो कितना स्ट्रांग है. ठीक है, if that bond is strong then it indicates कि it is past infection. ठीक है, if that bond is weak then it indicates weak infection. Okay. So this is known as IgG ability test. So IgG ability low means it is current or acute infection. ठीक है, अगर IgG एंटीबॉडी का टाइटर इंक्रीज हो रहा है पेयर्ड एंटी सीरम, देन अगेन इट वुड इंडिकेट इट इज recent infection. Old infection होगा तो IgG levels would be stable throughout. ठीक है, IgG antibodies alone without IgM या फिर high IgG ability would mean past infection or vaccination, and it denotes immunity. So IgG अगर मिल रही है against rubella in any mother, then the mother is not at risk. That means she is immunized. ठीक है, congenital rubella fetus में देखना है या न्यूबॉर्न बेबी में देखना है तो IgM एंटीबॉडीज कर सकते हैं या फिर ये आपको कल्चर दवाइज. ठीक है, so prophylaxis के लिए we have this MMR vaccine which is now used, that uses RA27/3 strain. Again, it is a live attenuated vaccine given along with measles and mumps. This vaccine is contraindicated in pregnancy because it is a live attenuated vaccine, and live attenuated vaccines are generally contraindicated during pregnancy or in immunocompromised patients. Okay, but again, before the discovery of this vaccine, the children who are suffering from rubella would be identified, and they would have parties. Okay, in that party, they would invite other children who had not yet been infected with rubella, in a hope that they will get infected. Primarily the girls, because they knew about the congenital rubella thing. Okay, so young girls would deliberately be infected with rubella so that they would develop long-term immunity. But again, this is not a good method or approved method, but still it was practiced sometimes. Now we do have a very good vaccine, that is the MMR vaccine, included in our national immunization schedule. Okay, so these were the important viruses that are capable of causing viral exanthematous lesions. So thank you. Any questions? All right, thank you.